Hi, my name is Olivier Gimenez. I'm a CNRS scientist at in Montpellier, in France. I'm going to tell you about my first steps in deep learning with R and the study of species interactions with camera traps. Okay, I work on predator-prey relationships, which are interactions between species that drive large mammal communities. And this is joint work with Anna Chen and Maëlys Kervelec, Vincent Miel, and colleagues from the French Office for Biodiversity, OFB, and game federations from Ain and Jura counties. So colleagues from OFB and game federations collect the data on species of interest with camera traps set up as strategic places on both sides of forest path, for example, where species tend to pass, like on this picture where we have a lynx and its kittens. This method is non-invasive. In other words, we do not need to physically capture the individuals, which is cool. The thing is that we get tons of pictures that we have to tag with a species label. And then this is where deep learning is very useful. The idea is to feed algorithm with pictures as inputs and get the species ID as outputs. For my analysis, I use the FastAI library, which relies on Python and its library PyTorch. The advantage of this library is that it comes with an R package that provides a bunch of functions to use it. Sorry. What are the results I got? First, I did some transfer learning on a study site in the Jura County where we had a ton of pictures manually tagged. To do so, we only need a few lines of code. We first load the FastAI library. Then we load the picture that we split into a training and a validation data set and apply all sorts of treatments to these pictures. We get the pre-trained model architecture, here ResNet 50. And then we continue training the model with our data. I used 20 epochs and saved the model after each epoch for later use. The results are rather good. We managed to classify the lynx and its prey, main prey, chamois and roe with a satisfying level of certainty. Then I used my freshly trained model to tag automatically a bunch of pictures we had on another study site in the Ain county. And these pictures were also manually tagged. Therefore, we knew the species with certainty. In R, we use the predict function, as usual, to tag a new picture with the model we've trained in previous step. The results are displayed in a confusion matrix with species that are predicted by the algorithm in columns and the species that are actually in the pictures in rows. A false negative is, for example, a picture with a lynx, but we predict another species on it. And a false positive, sorry, is a picture without a lynx, but we predict that there is actually a lynx on it. So it's a false positive. Starting with false negatives, here in rows, for the lynx, for example, we see that we confuse lynx with, for example, cats. And now with false positives in columns, we predict lynx on picture where we have actually a roe deer. Okay, so this is a bit disappointing, but past the frustration, one may ask, is it really a big problem? Will our answer to the ecological question of predator-prey interactions be affected by these errors? And to assess the bias, we use the statistical models that allow inferring co-occurrences between species while accounting for imperfect detection. These are the so-called occupancy models. And you can fit these models in R, which is cool, with this package unmarked. This is a snapshot of the data, one panel per species, so lynx here, fox and cats, and with camera traps in rows and time in column, sampling occasions in columns. Detections are in blue and non-detections in pink. With this data, we build models to test our hypothesis about interactions. And here we look only into interactions between lynx and its prey. And we also consider species-specific detection probabilities. And we fit our model to the data using maximum likelihood. And then we get occupancy probabilities for lynx given presence or absence of its prey. In red, we have the estimates from the model fitted to the picture that were manually tagged, and in other words, without false negatives or false positives. It's the ground truth. In blue-gray, the same model fitted to the picture that were automatically tagged by our trained model with a CNN, convolutional neural networks. And there is a small bias here in the occupancy probability of links given the presence of both its prey when we rely on occupancy or on automatic tagging. Okay, the difference in estimates is small, and we may well decide to ignore this bias and save us a lot of time by not tagging picture manually. Now we have a bigger issue here with the occupancy probability for links given roe deer presence and chamois absence, which is clearly underestimated. Okay, to conclude, training a model on one site and predict on another site is difficult. This is a non-result. Our problem is most likely due to a lack of diversity in the training dataset, and we are in the process of adding more pictures, more diverse, and we do the training. And it's easy to get lost in deep learning and lose sight of the ecological question. And if I can live with small bias on ecological indicators, it's better to stay on course and go with not too perfect performances of the algorithm and save precious time in tagging. And thank you for listening.